Hello and welcome to this PIR Live Event webinar, brought to you in part by Partners in Research Canada. My name is Ben Hobbs, and I will be your host today. If you're joining us live, remember that you can ask questions at any point using the Q&A, which is located near the bottom center of your screen, and type your questions into the pop-up window. I will relay these questions to our guests during the Q&A portion of the webinar. So without further ado, I would like to introduce our guest today, Dr. Gosia Korbas, Senior Scientist at the Canadian Light Source and Adjunct Professor in the Department of Anatomy and Cell Biology at the University of Saskatchewan. Thank you for joining us, Dr. Korbas. I'll let you take it from here.
Hello, can you guys actually see me? Because I can't see myself. So
yeah, we can see you. It's perfect up there now.
Okay. Hello, everyone, and welcome to BioXAS sector at the Canadian Light Source. I'm so happy that you guys could actually join us for this live webinar. And the plan is following. So I will be giving you a short presentation on how synchrotron can actually help scientists in their research. And I will use two examples from my previous research that I've done on Mercury, which is my favorite element. And then we'll take you on a tour of BioXAS sector. So let's start. This is Canadian Light Source that you see on the picture. This big building in the center is actually located on the grounds of University of Saskatchewan in Saskatoon. The size of the building, the footprint of it, is close to the football field. So it is pretty big building inside. We have 22 beamlines. A couple of them are still under construction and commissioning. And we are actually finishing our work on BioXAS sector. Almost 1,000 scientists comes every year here to Canadian Light Source to conduct their experiments, and they come from various places around the globe. They are interested in different research in health, in agriculture, environment, and material science as well. And this is how Canadian Light Source looks inside. There is actually a mezzanine level here. I'm trying to point with my cursor. And when you come for a tour, actually, this is where you would be walking around. You can look down and you can see a round structure in the center. This is storage ring. And this is actually where we accelerate. We keep the electrons there and they produce the light. This light, so that's the round structure here, more or less. And this pink arrow here is actually where BioXAS is. So the light now comes from the storage ring, is captured here and directed into the end station where the experiment is done. You can actually see these white walls here, the hatch, and this is where we actually go for our tour after this presentation is done. This is inside of this white hatch that you just saw on the previous picture. This is where we'll go, and I will explain a little bit how the beamline works and how we produce the light and what we do with the light. BioXAS is actually comprised of 3 beamlines, a quite unique facility here at the Canadian Light Source. Those beamlines are dedicated to biological, environmental, and health-related studies, and we have 3 beamlines here. So 2 beamlines are for X-ray absorption spectroscopy technique, and 1 beamline, which I mostly work on, is BioXAS imaging, and this one is doing X-ray fluorescence imaging. I think the best part for me to work at the Canadian Light Source is actually the fact that I can interact with so many different scientists, technicians, engineers, IT support people, business people, administration staff here. And this is really what makes this work very, very exciting. You can see on this picture my team, so the team that I usually work on day-to-day basis. So from the left, you have Sean and David. These are 2 engineers that tremendously help on the BioXAS project. You can see me very excited, and next to me is my support scientist, E.B. Bondici. This is from January 2017. We are celebrating at that time a major milestone for BioXAS imaging beamline. This is when we were able to deliver the beam down to the last hatch. And we are definitely happy. You can see that I was very happy as well. So now back to the techniques, because I will be talking about the techniques first, but then I will use examples from my research in the past to illustrate how they can actually help and what sort of questions they can answer. So we have X-ray absorption spectroscopy, which determines chemical form of metals and other elements of interest in the sample. We call it speciation. And in a nutshell, the experiment is done the following way. So we measure the intensity of the beam that it's before the sample and after the sample, and we repeat the same measurement for a series of energies, and this is how we build what we call absorbance pattern, shown on this graph. The shape of this pattern is very unique. It's like a fingerprint of a certain chemical present in your sample. Samples that we can measure using this technique are many. It could be intact biological tissues, it could be soil, proteins, in principle anything that you can stick in the beam and you are interested in looking what sort of chemical species of a certain element are present in it. Another technique, X-ray fluorescence imaging. Here we are not producing graphs like absorbance patterns, we are here producing images of elements distribution in a sample. And sample could be, again, organ tissue sections, could be plants, could be cell cultures. Sometimes people also put really big paintings or even fossils. It really depends on how beamline is equipped. And the experiment is done the following way. You usually need to focus your beam. So X-ray beam that comes, we make it even smaller. And then we move the sample in this beam and we build the image pixel by pixel. And the data for each pixel are collected by this X-ray detector. And of course, there is an electronics and computers that analyze those data. And at the end, you will get images of those different elements in the sample. I will show you later on how we can use this technique to image mercury in the tissues. So those two techniques, X-ray absorption spectroscopy and X-ray fluorescence imaging, are really great techniques to look at elements, and elements that are important from biology point of view. We can classify those elements as beneficial, and you heard probably about cobalt, iron, zinc, copper, molybdenum, selenium monomanganese. These are beneficial to our health. However, there are some elements that are not so. They are toxic, actually. You probably heard about arsenic and lead and thallium, mercury, and actually, surprisingly, probably selenium as well, because selenium is one of those elements that, depending on its chemical speciation and concentration, it can be both, actually. So it can be beneficial, but it could also be toxic. And this is pretty old concept. The first person that came up with it, the author of it, was Paracelsus. He was a Swiss medical doctor. He was a free thinker of his time, so he definitely faced lots of controversy in the 15th, 16th century. And he's considered these days as a founder of modern toxicology. And he was the one that first said that it's the dose that makes compound toxic. So now let's move to my favorite element, and I really like mercury. It's a very toxic element, but it's a very intriguing element. It naturally occurs in the Earth crust. It is released to atmosphere during volcano eruptions, but also coal burning and something what is called small-scale gold mining. I don't really have much time to go into details of small-scale gold mining, but this is a really important issue these days. Because of that, we see increasing levels of mercury in the atmosphere since basically Industrial Revolution. The common ore of mercury that occurs around its cinnabar, and it was used by ancient people. It has a beautiful bright red color. So when you Google, for example, cinnabar, you will see many, many different pieces of jewelry. The most toxic form of mercury is what we call organic form, because we have carbon covalently bound to mercury atom. And this is the compound that is present in fish and seafood products. And I will get back to it in a moment. Paracelsus, that I mentioned before, he was actually the first one who formulated a treatment for syphilis. And unfortunately, he did use mercury as a compound, active compound in his ointment. So he was treating syphilis, but unfortunately, at the same time, he was poisoning those patients. You may also have heard about a phrase met as a hatter, or actually no Mad Hatter from Alice in Wonderland story. This term was coined in the 1800s because hatters at that time, in the process of making felt hats, they were using mercury's nitrate. And this compound, you know, when it is toxic compound, and especially when they were working in a confined spaces, they were inhaling lots of vapors as well of it. This is what made them sick, and that's why when this term came to life. Recently, mercury was still used in thermometers, switches, thermostats, batteries, fluorescent lamps, in certain countries also in skin lightening products and traditional medicines. However, there is a global effort to phase out mercury from this product, and this is where Minamata Convention on Mercury comes. It came into force in 2017, and this is a global treaty signed by many countries worldwide to protect human health and environment as well from adverse effects of mercury. When it comes to speciation, it's definitely very important for mercury. It's not really the mercury atom itself that it's toxic. It's really its toxicity is defined by what it is bound to. In case of organic mercury, so the first compound shown here, methyl mercury cystanate, we call it. So there is a mercury bound to methyl group, and on the other side, it's bound to cysteine amino acid. This is exactly the form that you will see in fish and seafood products. And what it makes it so toxic is that it is readily absorbed in the gastrointestinal tract. More than 95% of it will get into your bloodstream. And unfortunately, it will trick our system in allowing it to cross the blood-brain and blood-placental barrier. So there are special cells, endothelial cells in the brain lining the blood vessels that their role is to protect the brain from harmful chemicals. Unfortunately, methyl mercury cystanate can sneak through them. And that's what it makes it so toxic. On the other end of the spectrum, we have the cinnabar that has been, you know, around for many, many years. It has been used in traditional Chinese medicines. It has been used also as a red dye in tattoos, as a tattoo dye, and only 0.2% of it is absorbed in the gastrointestinal tract. Overall, its neurotoxic potency is estimated to be 1,000 times less than methyl mercury. So it's definitely way less toxic than the other form, but they both contain mercury. So now let's move to our examples. So the first one is the zebrafish example, and in this research, we use X-ray fluorescence imaging. We were interested in finding out what are the differences in uptake of inorganic and organic mercury. So organic is the one that I said it's bound to methyl group, and the inorganic one, in this case, we use mercury chloride, which is a contaminant that sometimes can be present in the environment as well. We wanted to see what are the differences, how the tissues react to those two compounds. So we exposed small zebrafish larvae. Zebrafish is a very popular model system in biology, especially in developmental biology. Zebrafish is the same fish that you can actually get in a pet store, but we were exposing not adults, we were exposing larvae, and they were more or less 3 to 5 days old. We spiked water with those compounds, and we looked at accumulation of mercury with X-ray fluorescence imaging at different areas. So first, here on the left, we were looking at the section through the fish nose, through the section in the section of the fish head, and the section of the fish gastrointestinal tract. And then here on the lower panel, there are images of mercury from the synchrotron, and on the top panel, these are histological images. So we always take two sections. One section remains unstained, and this one goes for the synchrotron. The other one we stain with certain dye just to show all the cells and tissues in a better way. Otherwise, it really looks transparent. The overall message here was that the levels of mercury in the fish that was exposed to organic mercury were significantly higher. You can definitely see it in the head section because dark blue means that there is no mercury. So look at this head section of the fish exposed to inorganic mercury. There is almost no mercury there. Whereas for organic mercury, there is quite a lot of mercury, and I will get back to this result in a moment. Then when we move to gastrointestinal tract, it's the same result. We still see a little bit of accumulation of mercury in the fish exposed to inorganic mercury, but way, way more in the fish that was exposed to organic mercury. And just to stress, the amount of mercury itself that the fish were exposed to was the same. It's 1 micromolar of either mercury chloride or methyl mercury chloride. So it's the same amount of atoms. However, the speciation resulted in different uptake. Interestingly, the fish nose, so actually here you can see these are epithelial cells lining the nose of the fish, and these ones are actually accumulating the same levels. So it didn't matter if it was organic or inorganic, you can still see the same levels of mercury in both, which told us that both of those compounds, when they accumulate in the fish nose, they may actually affect sense of smell in the fish. So now back to this really interesting head section from the fish that was exposed to organic mercury. It was the first time that we see those round structures. And what it is, this is actually the island. So we could see mercury accumulating, organic mercury accumulating around the islands. And then also a little bit of more mercury than in other tissues in the retina. However, when we collected those images, we didn't have access to very small beam spot size, because you really want small sizes. The smaller you go with your beam, the more you can resolve. So we had to take those samples to a different beam line, and this one had a smaller beam spot size, and here we could actually see where this mercury exactly was localizing. So we are looking again at the head section, just stained with a different stain, so that's why it looked pinkish here and not blue. And then we are looking at this rectangular section here, stained section here, and then unstained. There was a little bit of lens here, and all the layers of the retina. When we look at the mercury distribution, beam was small enough, so we could definitely see mercury accumulating in this layer. And these are actually photoreceptor cells. So with help of a synchrotron, we could determine exact location of mercury in those fish. And this is really, it resulted in a really nice image and really lots of information as well. So you can see that when we plot all those three elements together, zinc is localizing in this black pigment here. It's called pigmented retinal epithelium. And then blue is mercury, so these are the photoreceptors. And then phosphorus localizing in cells because due to DNA in cell nuclei. So this research really showed that organic mercury is a potent neurotoxicant. It does accumulate in the central nervous system, but it also accumulates in the sensory organs. So its toxic action could be also by direct accumulation in those. And I have this problem. Okay, now it's accumulated. Sorry, some technical issue. So now let's move to the other example, human brain. We are interested in determining what happens with mercury. So we know through the bloodstream it will get into the brain as this organic mercury, but then what exactly happens to it? We had access to 2 samples of human brain from accidental poisoning cases that happened in the past. Case 1 was a female that was exposed to dimethyl mercury. This is the most toxic organic form of mercury that you could and you cannot really encounter. This is more like in a research laboratory that you could work with. And she actually did poison herself by skin contact with this compound, and she died 10 months after the exposure. Case 2 was also a female. It was 8-year-old girl who consumed contaminated pork, and she survived for 21 years in a debilitated state. So these were 2 cases when we had a really short time for methyl mercury to possibly change, and way longer time for this methyl mercury to actually convert maybe to something else. So that was basically the question: what happened? Can we see differences? In this research, we use Xray absorption spectroscopy. So The approach here is that we have this absorbance pattern of the human brain, and then we measure absorbance patterns of different model compounds. So we exactly know what the species of mercury are in it, and then we then try by linear combination to reproduce the shape of the absorbance pattern of the human brain with those of the model compounds. And in this case, for the case 1, so this 10 months between the poisoning event and the death, we are able to see those 3 compounds present. So methyl mercury cystanate, so the original form that was uptaken by the brain, mercury bistiolate, so another inorganic mercury form. And this was really very interesting result at that time. It was a mercury selenide, but in the form of nanoparticles. And this is actually my last slide. So we compare those 2 cases. So in case of 10 months exposure, as I mentioned in the past slide, so we saw original form that was accumulating, but we also saw already presence of other forms. And those forms must have been made from that methyl mercury. And this was consistent with what we saw from the other case. So when it was this long time between the poisoning event and the death, the original form, methyl mercury, was gone. We couldn't really see it in the brain anymore. However, we saw the other 2 forms, and quite high proportion of this inorganic mercury was actually in the form of nanoparticles of mercury selenide. And as usual in research, you know, you study something and then you think you answered the question you had, but actually this creates even more questions for you. So the question was here actually that, okay, we see this change, it changes to inorganic mercury, mercury selenide, but actually now the form is nanoparticles. Is it actually okay? Is it doing something in the brain? And with these open questions, I will end my presentation, and we'll now go for a tour of the BioXAS. However, if you are interested and you want to talk to me later on, this is my website, and please contact me. I will be happy to answer any questions you may have. So let's now go for a tour. Stop sharing. And I need to stop sharing, yes. So I do this here, or maybe we ask, oh, stop sharing, sorry. So let's now go for the tour. So please follow me here. Okay, so this is this room that you saw on one of the slides, and this houses all the optical components for all 3 beamlines. You see many mirror tanks, monochromators, and I will explain in a moment what they are. But maybe let's start from the beginning. Let's start where actually the beam enters this hatch. So here behind that wall, there is actually storage ring. This is where the light is made. And then the light, this here, undisturbed. It's basically just light produced by the storage ring comes here. And then we filter out from this beam that comes from the synchrotron 3 separate beams. That's why you see actually here 3 different beam types. So there's 1 and 2 and 3. And actually the one that is the closest to me, this one belongs to BioXAS imaging beamline, and we'll just follow the path of this beam, and we'll follow as well the light path. Here we actually have a place where we have diamond filters, and we use those diamond filters to filter out portion of radiation that we don't need. And you can actually see that the light that comes is quite powerful. It browns the glass on the viewport here. So let's now follow, and then here you can actually see that when we open, you may see, you may not really see, inside we have those diamond filters. And actually what happened was that normally it should have been closed, but a couple of times we forgot. And we left it open, and that's why the glass got brown. So I close it now. So let's follow the beam path. So now the beam for the BioXAS imaging would just pass through some mirror tanks, but it's not interacting with those mirrors. The mirrors are actually for the other beamlines. So the beam is just passing through, not interacting with anything up to really here. So this is the first mirror, and the mirror here we're talking about 1 meter long silicon slab covered with a rhodium coating. And this is a mirror that is downbound, so it means that as the beam comes, it actually reflects the beam down. And the beam is still quite powerful here. That's why you can see this browning on the glass slide, but only half of it, because the mirror is actually positioned this way. And the light comes down and scatters this way as well. It doesn't scatter that way because it's blocked. So now let's follow X-rays again. So another beam pipe here, and this is big monochromatic. That's quite a big structure here. And this is for us to really only take a very narrow energy range. So up to this point, we have quite broad energy range of X-rays, but we don't really need it for most of our experiments. So we use the monochromator to just take a small portion out of it. And this is not a constant setup. We can actually change it. So depending what we are interested in, what elements we are interested in, we'll change this tuning of this monochromator. So now you can definitely see here that the beam is not so powerful because the viewport is completely transparent. It's just the pure normal glass. So this is another mirror, reflects the beam up, and then there is just the beam pipe, and the light travels here up to another end station, down to the imaging end station, and this is where we are going right now. So now we are back to where we started. This is where normally users, scientists would be sitting at the computer. That's right. And doing the experiments, analyzing the data, collecting the data. But actual experiment is done here. So the beam pipe that I was showing you in the other hatch comes through this wall here. And then at the very end of it, we have small apertures. So we make the beam that comes from the other hatch even smaller. And the experiment is done in the following way. Pointed again. Okay. So here we have this. These are the apertures here. And the experiment is done the following way. So basically, yes, we move the sample around, and the sample in this case are the set of 3 dry leaves. We're actually testing the stage not so long ago, and that's what we used for testing. Very useful, nice sample actually, dry leaves. So now we can head back to a computer, and I can show you what sort of data we're actually getting from those leaves. So this is our data collection software, and you normally would be sitting here, you would set up your experiment, and eventually this is what you would see. So blue, it means that it's a very low concentration, and red means very high. And so this is an image of a distribution of iron in the sample. So naturally, there is no iron outside the leaves, but there is quite a lot of iron inside the leaves. And this is the X-ray spectrum. So these are the data that come from the synchrotron, and I can only choose this peak because I know that that peak at this energy belongs to iron. I can also look at another element, for example, zinc. So I can tell the program that only look at data coming in from this peak, and then build an image of zinc, zinc distribution. So basically, this is how we collect the data on BioXAS imaging beamline, and I think we'll end here. Thank you so much.
Well, thank you, Dr. Korbas. Just before we move into the Q&A, I'd like to remind those classrooms joining us live, they can direct their questions to the Q&A, which is located in the bottom center of their screen. So our first question today comes from the grade 11 students in Mr. Hickey's classroom, and they were wondering what makes a metal toxic or beneficial?
It really comes, I think, to, to processes. So it's funny actually, because I was thinking about this question today when I was taking a shower, honestly. I was thinking that it could be one of the questions that somebody can ask. I'm not really, biologist by training, but from what I know, I think it's a matter of how our bodies are utilizing those elements. So copper, for example, there are many processes, biological processes that use copper, and our organisms know how to deal with excess of copper. Mercury, for example, our body doesn't really know exactly what to do with it. It will, it will accumulate it inside, and it really triggers a series of, events inside cells that are not really beneficial. They are not good. They are actually damaging to the cells. And that's basically where it is. I think it's a matter of, our organism being able to handle and to make it less toxic. So also, for example, speciation is so important, yeah, because even with copper, it is beneficial as an essential element. But, there are certain diseases in which our organism is losing its ability to handle excess of copper or excess of iron, and this will make people not toxic. It will make people sick because it's so toxic. So it's really the mechanisms, that biological mechanism processes that are inside our bodies, that we know, our organs know how to deal with those elements.
The students from Louis Riel School in Calgary are wondering what are the main contributors to marine bioaccumulation of mercury?
Marine bioaccumulation. I'm not my marine biologist, so do they mean how mercury gets into the oceans, I guess, yes?
Yes, I believe so.
Yeah, so I haven't mentioned about this because it would take too long in another slide. So there is something what is called global cycle of mercury. So as I mentioned, mercury is released into atmosphere. From the atmosphere through the rain, for example, it will actually get deposited in the water. And then in water, there are microorganisms that can convert inorganic mercury to organic mercury. And organic mercury is more bioavailable. It's more readily absorbed, as you actually could see on the on the zebrafish research. Yeah. So this is how it gets into the marine life, basically.
The students in Mrs. Moore's class are wondering why you use different stains on different tests. I think you mentioned 2 different stains you used in your presentation.
Yeah, so yeah, one it's called methylene blue, and another we call H&E. It's in histology. There are many different dyes depending on what sort of cells or compounds you want to visualize. I would say that methylene blue and H&E, which is hematoxylin and eosin, are quite similar. It's really a matter of color. Initially in the lab we were using methylene blue because it was way easier to produce sections. You just dip it into the dye and take it out and wash it, and it's done. But H&E, to us, it seemed like it produced nicer images with better clarity, and that's why we ended up using H&E. So it's really a matter of choice and what sort of tissue you are working with.
How large of a sample can you fit into the sensor?
So that big stage that you actually saw over there, it can handle quite a big sample. So, you could probably put like a slice of human brain, for example. I operate on centimeters and not inches. So I would say, something probably that is like 40 by 40 centimeters, at least, it would still we will be able to fit. Sometimes it's also a matter of weight, of course, because the stages, it's not only how far they move, but also what sort of weight they can carry. And I mentioned fossils, for example, so some of them are pretty heavy, or some paintings. So it really depends, but on BioXAS, I would say yes, like a slice of human brain, for example.
Mr. Stanlick's class is wondering what happens if mercury passes through that blood-brain barrier.
So when it passes through the blood-brain barrier, it's still in this form of methyl mercury sustainate. And this is part that we don't really know. So it accumulates inside the cells. And then with time, there are processes that will change this to this mercuric selenide form. And we suspect that this other form that we saw, so this mercury bistiolate that was present, it could be an intermediate form, but we don't know this. So we don't really know, we don't exactly understand what's the chemistry behind methyl mercury losing methyl group. So that's really an open question here.
The students from Calgary are wondering if small-scale gold mining contribute to mercury in the environment opposed to larger-scale mining operations.
Yes, so there is a big difference here. So that's why we don't talk about large-scale mining, but small-scale mining. So to clarify, so small-scale, or we call it sometimes artisanal as well, it means that on small scale, and people in villages somewhere in Africa, for example, they will use mercury to extract gold from sediments, and they will do it in a very uncontrolled way, because by mixing liquid mercury, which is very toxic, the vapors are toxic of liquid mercury because they can get into your brain. So they mix basically in their houses or outside in a shed. They will mix gold, sediment that has gold with mercury. It will form amalgam, which then they can wash from the sediment. And then in order to extract gold, they will burn it. They will heat it up, and mercury is released as a vapor. So this is really serious concern because they are using liquid mercury. They are not protecting themselves, and they are not protecting, of course, the environment. Whatever it's left, it will be discarded, and when it's heat up, it is released into the atmosphere. So there are global efforts these days to really reduce, to educate people as well, and to improve their lives.
Well, that's all the time we have for today. But before we end, I was hoping to have you speak a little bit about your career path and maybe some advice for students who are watching today and considering something similar.
So my advice would be just, I heard it somewhere else, honestly. It's follow your heart. I think that if you find something that you are really interested in, it really will help. It will keep you going, because it's not always rosy. There are problems and you struggle, long nights and exams. In my case, I'm not originally from Canada. I actually came to Canada in 2005. I'm from Poland. This is where I got my degree in physics, master degree, and then PhD as well. But from the very beginning, I was trying to combine many fields, and I think it's not really just physics. It's you basically just try to learn different things, and with PhD, of course, it helps to become a scientist. These days you quite often need that title and that further education to succeed.
Well, that's all the time we have. Thank you, Dr. Korbas, for taking the time to show your facilities to us and for answering all of our questions.
Thank you so much for joining me. It was a pleasure.
Next time on PIR Live Event, we are talking vaccines with Dr. Wilson from the Ottawa Hospital. More information about these webinars and other PIR educational programs are available at pirweb.org. Thanks for tuning in and have a wonderful day.